Namaskar and welcome to Instinctive Biology. Today we are going to see bacterial gene regulation with the help of lactose operon. Yes, bacteria can control their genes. Because of the environmental changes, they have so flexible that they can control their genes. So what would be the learning outcome for us today? We will in detail see the lactose operon, the induction, catabolite repression, concept of partial diploid, and this is very important from the CSIR NET exam point of view. So let us study that. Constitutive genes and inducible genes. Constitutive genes are continuously expressed inside our cells. They are the housekeeping genes. For example, glucose utilization genes, that is glycolysis genes, are always available inside a bacteria. They don't need any switch. So bacteria utilizes the glucose. But what happens when the environment becomes unfavorable for them? Glucose is not available. They utilize other sugars like maltose, lactose present in the medium or present in the environment. So according to the environment, the bacteria have to change their gene expression pattern. So in response to the environment, they alter their biochemistry but without compromising with energy efficiency, because they synthesize things only when they are required, and that is what is gene regulation. Now, what do we mean by an inducible gene? Inducible genes are expressed, that is converted into a protein, only when we have an external stimulus. That external stimulus is binding on the controlling side. Promoter element is the element where the RNA polymerase binds. All these elements are present on the DNA, that is part of the gene. Coding sequence is producing the protein by transcription translation, and terminator ends the gene. Now, what do we mean by positive regulation? Positive regulation means whenever a inducing stimulus is present, the gene is on, that is the switch is on. That means the gene is converted into a protein. So activator is the positive regulator. It binds to the region called as operator and promoter, and then RNA polymerase causes transcription. If activator is not there, there is no transcription, so no translation. So activator as well as the other component, repressor, should be able to recognize the appropriate condition for their action. And how do they do that? So they also have a switch in them. Activators are proteins. They are DNA binding protein. So they have two sites, allosteric site where the effector molecule binds. So no effector, the activator is not activated, it cannot bind to the DNA, no transcription. Effector is present, it binds to the allosteric site, and now activator has a change in shape and it binds to the DNA and there is transcription. Same is for repressor, and it causes negative regulation. Means repressor is present, it binds to the DNA and there is no transcription. If repressor is absent, there is transcription. Same way, repressor is also a protein which has two sites, one for binding of effector molecule and another where it binds to the DNA. All these are DNA and DNA protein interactions. Now this is the structure of lactose operon of E.coli. A very easy way to remember the sequence is PIPO-ZYA. P, PIPO, that is P, promoter for lactose, I, the repressor gene of lactose, again P, promoter for the ZYA genes. ZYA are structural genes standing for beta-galactosidase, permease and trans-acetylase, and regulatory genes are I and O, which control the structural genes. Now, what are these genes exactly doing? This is a bacterial cell, and we have lactose outside the cell. The role of permease when present is to bring the lactose inside the cell. Beta-galactosidase splits the lactose into glucose and galactose. Beta-galactosidase also converts the lactose into another form called as allolactose. Actually, allolactose is the inducer of the operon. So when allolactose is present, the operon works very efficiently. So this is very contradictory. How come lactose is coming inside without the operon being on? So always in any gene expression there is a basal level expression. So very small amounts of lactose operon enzymes are always present in the cell. But when lactose is getting converted into allolactose, that actually being the inducer causes high amount of gene expression. Now what happens in the absence of lactose? In absence of lactose, the I gene is producing a repressor. This repressor is going and binding to the region where RNA polymerase has to bind. So now RNA polymerase cannot bind because its region is occupied and there is no transcription and translation of genes. On the other hand, when lactose is present, what is happening is the repressor protein is produced, but the repressor protein is bound by our inducer allolactose. This causes change in the conformation, and so RNA polymerase binds to the promoter region, causes transcription and translation of genes. Now let us see what is the situation when glucose comes into the picture. So this condition is when glucose is very low. When glucose is low inside a bacterial cell, cyclic AMP levels are very high. We have another protein, catabolite activator protein, which binds to the cyclic AMP molecule, and this complex binds to the operon upstream to the promoter of lac genes. Now RNA polymerase can very efficiently bind and cause transcription and translation. Why? Because in the diagram we can see that binding of cap-cam complex bends the DNA, and this escalates transcription rate, and so we have high amount of beta-galactosidase permease and trans-acetylase being produced. But what is the situation when glucose is very high? When glucose is high, cyclic AMP is very low, so it does not bind with cap, and RNA polymerase does a very low level or no expression of the lactose operon gene. So the presence of and absence of glucose also matters a lot in the lactose operon. These are the two pioneering scientists, Jacob and Monod, who have proposed the lactose operon model in 1961, and in 1965 they had received a Nobel Prize for their innovative work. Now what did they do exactly? They produced partial diploid cells. They took bacterial cells, did conjugation experiments in them. In this conjugation experiments they used a plasmid, and this plasmid had a lac operon gene. So what generated was a partially diploid cell. Partially diploid means carrying two copies of lactose operon gene, and in this way they discovered mutations of structural and regulatory genes, lac I plus, lac I, OC minus, and from this they proposed the operon model. Now this is a bacterial cell which is a partially diploid E.coli strain. Here we can see two lactose operon copies, one of E.coli itself and another from the plasmid. So if we see the genotype contains lac I plus, O plus, Z minus and Y plus from E. coli cell, and from the plasmid we have O as a constitutive expression. Why? Because there is a mutation in O producing OC, and this mutation causes it to unable to recognize the repressor, sorry. So when I plus produces the repressor, it goes and binds to both the operator region, but the mutant OC unable to recognize the repressor and causes constitutive expression of genes of the operon, beta-galactosidase permease and transacetylase. So in the genotype of E.coli with such a gene sequence, there is always constitutive expression of the operon. Now we see another example where we have a partially diploid cell with the genotype lac I plus, O plus, Z minus, lac Y plus. In this case, the cell produces the repressor and it goes and binds to the operator region, but the F plasmid contains a mutant lac-I gene which produces a mutant repressor protein which is unable to bind to the O plus region. But as the lac-I plus gene is a trans-dominant gene, means its product bind to the regions of DNA apart from itself also, so there is no transcription and translation in both the cases and the operon is off. These genotypes are very essential for us to solve questions in CSIR NET exam. So in the screen I have given a question which you have to solve after watching this video. So thank you so much for watching and please do subscribe my channel.